Welcome everyone to my channel BioWorld once again. So after C3 cycle and C4 cycle, I've come up with photorespiration. Again, a small brief part which is also given in NCERT but not in a broad way, but it's an important part. Like you need to understand what is photorespiration. Now, remember, before getting into the cycle, it is not so useful process for the plant. It is a waste process. First point. Second point, it only takes place in C3 plant. That is, it only takes place in C3 plant but not in C4 plant. So, and what is the characteristic feature of this plant which undergo photorespiration? The condition of the atmosphere should be very high light intensity, hot temperature should be high. And so what happened during that period of time, the stomata remains closed or partially closed, which do not allow the entry of carbon dioxide into the plant. So concentration of oxygen rises in the plant. Remember, this enzyme is very important in Calvin cycle, that is Rubisco, which helps in combining carbon dioxide with RUBP in Calvin cycle. So this enzyme has an affinity to both of them, that is oxygen as well as carbon dioxide. So an enzyme has one single active site. That site can bind with either oxygen or carbon dioxide. When it binds with carbon dioxide, it undergoes Calvin cycle. When it binds with oxygen, it undergoes photorespiration. So what we have come to know, that when CO2 concentration is less in compared to oxygen, the Rubisco start combining with oxygen, and rather than entering into Calvin cycle, it starts the process of photorespiration for the plant, where no glucose is synthesized, rather there is utilization of energy. So it's a waste process for the plant. And it is also, the other name for photorespiration is PCO cycle, that is photosynthetic carbon oxidation. Remember, in Calvin cycle there was reduction going on, like carbon dioxide was reduced to glucose, but here it is not happening. Here is oxygen has been added, so it's an oxidation process. It is also named as C2 cycle, as first stable product is carbon 2 compound, and also known as glycolate metabolism cycle. So these are the names of the photorespiration, and it needs 3 organelles for this process. One is chloroplast, second is peroxisome, and third is mitochondria. Now, how the process takes place, how the cycle takes place, we'll start with the first one, that is, first is chloroplast. We know chloroplast is a place where light reaction takes place, and Rubisco is present over here. So what happened? Ribulose bisphosphate, RUBP, that is in presence of enzyme Rubisco, rather than combining with CO2, it combines with oxygen, and it forms phosphoglycolate. So this is glycolate metabolism cycle, where 2 carbon compound is formed, hence the name C2 cycle, along with release of 1 molecule of 3-PGA. This PGA will enter Calvin cycle. So the again one rather than there was 2 molecule of PGA, here only 1 molecule of PGA takes place. So that is also kind of waste. So after phosphoglycolate, 1 inorganic phosphate will come out and forms glycolate, and this glycolate will now enter the second cell organelle called peroxisome. Now, peroxisome helps in peroxide metabolism, we know it. So, from glycolate, 1 molecule of peroxide, that is hydrogen peroxide, will come out and forms an amino acid called glycine. You must remember, glycine is the simplest amino acid. So, this glycine now will enter the third cell organelle called mitochondria. There, this glycine will lose 1 molecule of carbon dioxide. So losing 1 molecule of carbon dioxide in mitochondria again will help in no process. Like if it would have been chloroplast, this carbon dioxide could enter Calvin cycle. But in mitochondria, as we know, no Calvin cycle takes place. So this carbon dioxide molecule will go waste. So from glycine, the next amino acid which will be formed is serine. Here the formation of NADPH will take place from NAD plus, DP plus. From serine, the serine enters the cycle goes back to the next cell organelle, that is peroxisome. From serine again glycerate will come back. From glycerate again regeneration of phosphoglycerate will give rise to RUBP, where 1 molecule of ATP will be used in releasing energy in this process. So glycerate will enter chloroplast for the regeneration of phosphoglycerate. Phosphoglycerate converts into RBP for the cyclical continuation. So in this way, the number of molecules which are formed passes from chloroplast to peroxisome to mitochondria, again from mitochondria to peroxisome, and back to the chloroplast. So in this whole process, what we have found that there was no production of glucose, rather there were utilization of ATP. That means energy was used here. That is a kind of waste. Secondly, one molecule of carbon dioxide was also released out in mitochondria, which would help plant in no way, as mitochondria is not the site of Calvin cycle or glucose formation. So overall, what we have come to understand that photorespiration is not so useful for process for the plant. It is a kind of waste process for the plant. So whenever there is high heat, high light intensity, oxygen concentration rises in carbon 3 plant, C3 plant, the rubesco starts combining with oxygen rather than combining with the CO2 and undergoes photorespiration, rather Calvin cycle, ultimately the resulting in the loss of energy for the plant. So this was photorespiration. In NCERT you will get a brief idea about this cycle and how this cycle takes place in the plant. Hopefully this video you would have understood in a proper way. Please do like, share and subscribe my channel. Thank you for watching it.